morning to you all. How's been the break? I hope you are learning from the comfort of your home at this moment. Please permit me to quickly encourage you and advise you to continue and be serious in your studies, in your learning, and try as much as possible to maintain the highest level of personal hygiene, especially in this period. We'll be discussing briefly on growth as a property of the cell. I remain my humble self, Sule Emmanuel O. Growth is one of the important properties and functions the cell of every living organism perform. It is an anabolic process leading to the building up of the body of living organisms. Anabolic process in the sense that it leads to building up of the body of living organism by the addition of protoplasm. Let's look at the definition of this term growth. Growth can be defined as the irreversible increase in size and mass, which we term volume, of an organism due to the addition of new protoplasm. This word protoplasm, or rather terminology in biology, protoplasm, simply means the cytoplasm together with its inclusion. It is the entire cytoplasm and the cell organelles in the cytoplasm all together make up the living component of the cell, which we term protoplasm. Basis of growth. Let's consider the basis of growth. 1. Cell division. Cell division has to do with splitting of a single cell so as to have 2 daughter cells or 4 daughter cells, as the case may be. And this cell division begins from the nucleus. And when the nucleus first of all split or divide to give rise to the division of formation of 2 daughter cells from one single cell parent, it's called cell division. And it is, because it begins from the nucleus, it is termed karyokinesis. And when it divides, when the cytoplasm constricts, it is termed cytokinesis. We'll see it later in our discussion. Number 2 basis of cell growth is cell elongation or cell enlargement. What do you mean by when the cell divides? The next thing is that cell becomes enlarged. As it enlarges, it means is becoming elongated. And the next basis of cell growth is cell maturation. When the cell divides and it becomes enlarged, the next thing is it becomes mature. And as it matures, it becomes differentiated, which we term cell differentiation, which means changing in their shapes or forms to perform certain functions. So the cell divides, it elongates or enlarge and become mature, and then become differentiated. And when it becomes differentiated, it changes in its form or its shape or its structure generally, so that it can perform certain function. How do we measure growth of an organism? Growth of an organism can be measured by using its mass and weight, length, height or width, area or volume, depending on the types of organisms under consideration. Some organisms are measured, the growth of an organism can be measured using its mass or weight. To some plants or some organisms, their growth can be measured by measuring their length. For instance, the growth of a maize plant can be measured or estimated using its height. So when we use the height of a maize plant to measure its growth, this same height of a maize plant cannot be used to measure or estimate the growth of a mouse. So we rather use the mass of the mouse to measure it or estimate its growth, depending on the organisms. Now let's consider this word mitosis, or terminology rather. Mitosis. Mitosis is a process by which a cell nucleus divides into two daughter nuclei containing identical sets of chromosomes of the parent cell. Mitosis is a continuous process which occurs in 5 stages as shown below. When we talk about mitosis, we're talking about the division of cell nucleus, first of all, which we termed karyokinesis. And then the cytoplasm also follows suit to constrict and divide to form 2 daughter cells. So the result of mitosis is 2 daughter cells, leading to the multiplication in terms of number or in terms of size of an organism. So let's consider these stages of cell division. One of the stages is interphase. Here, nothing really happens. It is usually called the resting phase, the period during which the cell is equipped, it becomes energized. So, with the appropriate energy needed to carry out synthesis of organelles and increase in size. The next phase is prophase. This is the longest phase. Majority of the activities of cell division take place here. And in this phase, prophase, each chromosome replicates and now exists as a pair. Another thing that happens during prophase is that the chromatid shortens and thickens. In other words, the chromosome become condensed. The spindle fibers is formed around the centriole. This centriole is an organelle inside the cell that is responsible for cell division. The nuclear membrane is still visible, is still intact. Let's look at the drawing, the picture of prophase stage of mitotic cell division. So from the diagram, you could observe that the nuclear membrane is still visible and very intact surrounding the chromosome that is condensing. Look at the nuclear membrane here surrounding the chromosome, and the chromosome is inside, is condensing. Now we have this centriole responsible for the cell division, and then we have the spindle fiber trying to be moved towards the opposite pole. Now let's consider the second phase of mitotic cell division, which is metaphase. Here the nuclear membrane disappears, so the nuclear membrane is no longer intact or visible around the chromosome. The chromatids now become attached to the spindle fiber at their centomere. So what do I mean by this centomere? Is the joining segment, this joining portion of the 2 chromatids to make it a pair of chromosome. And then the chromosome line up at the middle of the cell. In other words, the chromosomes are aligned at the equator of the cell. That's one of the major features of this particular phase of cell division, especially in mitosis. So when you see the chromosomes become aligned at the center, that is at the equator of the cell, that is called the metaphase. Now let's recognize the diagram from this picture. So you can observe from here that the chromosome, look at it, they are aligned at the middle of the cell. That's what I mean by this equator of the cell. And then you can see that the nuclear membrane is no longer there. You can't actually observe any nucleus there, not to talk of the nuclear membrane, except the plasma membrane surrounding the entire cell, right? So you can see the spindle fiber shooting out to become attached to the chromatid at the equator of the cell. So as soon as they contract, they will start pulling it apart. Now let's go to the next phase of mitotic cell division, which is the Anaphase. Excuse me. Here, the nuclear membrane disappears, and the spindle fiber pull the sister chromatids to the opposite ends, that is, the opposite poles of the cell, as the spindle fibers contract. Now let's look at the picture. Now, in this picture, we could observe the centriole here. Look at the centriole here. And then this is what I mean by the spindle fibers. Those that shoot out from the centriole to become attached to the chromatids. These are the chromatids. Now, at this point in this picture, you can observe that there is a wide space between the sister chromatids at the center of the cell. And you could observe also that they are being pulled apart. Look at this line going towards this pole, and this line also going towards this pole. Now, in other words, the major feature of this particular phase of mitotic cell division is that the spindle fiber become attached. As they become attached, they start to contract, and as they contract, they pull apart the chromatids at the center towards the opposite pole. So when you see this picture, you will immediately recognize the anaphase of the mitotic cell division. Now let's proceed to the next phase of the stages of mitosis, which is what? Telophase. Here, the nuclear membrane reappear. It's trying to be formed again, and the chromatid reach the end poles. So look at the items there. Is the major thing that happened during the telophase. The spindle fibers disintegrate, they disappear, and nuclear membrane forms around the chromatids. The nuclei they reappear again, and the cytoplasms divide. So what do I mean by the division of the cytoplasm? I mean, in a word called cytokinesis. So the moment we get to the telophase, the cytoplasm constricts at the middle, at the center of the cell, trying to divide to give rise to two daughter cells. And this is what we term cytokinesis, to give rise to two daughter cells. Let's check the picture. Look at the cell membrane intact there. And then the nuclear membrane reappears again, surrounding each group, each set of the chromosome. And then you can see that the constriction of the cytoplasm at this point, the cytoplasm constrict here, at the center of this single cell, so that it can give rise to two daughter cells. So the result of these stages of mitosis is two identical or two daughter cells. And I want to believe you learned a lot from this discussion of ours, and I want to give you this assignment. Try as much as possible to do justice to this assignment. You have to state 4 ways by which plant growth can regulate plant hormones. State 4 ways by which plant growth can be regulated by plant hormones. Please send the answer to this number on WhatsApp, 07066584960. Please, once again, send the answer to 07066584960. Thank you.